can pull that up real quick again. So you can see here my cyclin concentrations are what are changing. So as the cyclin is degraded in the cell, its concentration is going to fall. Okay? And so the cell has already moved forward, and so that cyclin is no longer needed. And when that cyclin gets degraded, it is released from the cyclin-dependent kinase, and so now the CDK is deactivated. It's not destroyed, it's just deactivated. Okay, so let's walk through a particular cyclin example. And I know it seems like we've just kind of said this over and over and over and over again. But on your test, it's always helpful to have examples that you can write about. And like I said, this is really the new stuff that you've never heard before. Okay, so let's look at this cyclin example here that's called M-cyclin. Okay, and so M-cyclin, remember the M-cyclin level, that's what's going to change. You can see that here in kind of this darker purple here. You can see the M-cyclin concentration rising and then falling, rising and then falling. And so that cyclin is going to bind to the CDK. So the M-cyclin binds to the cyclin-dependent kinase. And so when that binds to the cyclin-dependent kinase, that becomes a complex called MPF. Okay, MPF stands for mitosis promotion factor or maturation promotion factor. Okay, but this MPF is going to it's going to help regulate mitosis. Okay, you can see here it comes in at the near the end of G2 into the M phase. Okay, and it's going to remove an inhibitory phosphate group. So it's activated at the end of G2, okay, and it's going to remove an inhibitory phosphate group. And when it removes that inhibitory phosphate group, then it allows the cell to move forward through mitosis. It's then able to phosphorylate target proteins at this particular point. Okay, and so this is going to promote the actual events of mitosis. And so you can see that when it's not in, that M-cyclin gets degraded, okay, and its concentration lowers significantly when it is not time to activate that MPF. Because remember that MPF, that CDK, is not what gets broken down. It's the actual cyclin. Another example would be SCDK. Okay, and so SCDK is done with cyclin A. Okay, sorry, I should probably write this better as CDK, which is done by cyclin E. Yeah, I'm sorry, cyclin B. And so the SCDK, which is done with cyclin A. So when cyclin A is present and binds with the CDK, this allows the cell to then initiate DNA replication. When that particular CDK is activated by the cyclin A, the cell can then initiate replication. Again, we're going to look at that graph again real quick. Okay. And so you can see where these cyclins are activated, right? Cyclin A concentrations are high, okay, here, and they fall after it's replicated, and then cyclin B is activated right there going into mitosis. Okay, its concentrations are high, and then it is degraded after that so that it will then deactivate. So in addition to those cyclins, the cell also has checkpoints that help control whether it's going to move forward or not in cell division. So the first one that we have here is what's called the G1 checkpoint, okay, also sometimes called the R point. So at the G1 checkpoint, this is the primary determining factor for whether the cell is going to go on and do cell division. If the cell can get past this G1 checkpoint, then it is most likely going to be able to continue on to do cell division. So it has what is called the R point that needs to be passed here. So once the R point is passed, then the cell is given the go-ahead. The cell is given the go-ahead signal to move on into the S phase, move on into that synthesis phase. Okay? And that R point could be due to cell growth, organelle reproduction, but this is the signal that the cell is essentially big enough, strong enough, ready to replicate the DNA. If the cell can't get past this signal, that's where it goes into, so it can't get past that signal, that's where it goes into that G0 phase, okay? And it will not go on and replicate. The next signal would be down here at the G2 checkpoint. Okay, and so the G2 checkpoint, this is going to be my commitment to actually start mitosis. Okay, so the G1 checkpoint is to get me into the S phase to be able to do replication. The G2 checkpoint is to commit to mitosis. And at the G2 checkpoint, the cell has made a check to ensure that the DNA was replicated correctly, so that it prevents damaged DNA. That cell won't go on and replicate and pass that on. Okay, and this is where that CDK and cyclin combination, the MPF, come into play. The MPF signal needs to be strong enough here to push the cell over into mitosis. The other checkpoint, the last checkpoint we have here, is the M checkpoint. And the M checkpoint is going to ensure proper chromatid separation. So this is happening during mitosis. Okay, and so this checkpoint here, like I said, is looking to make sure that the chromatids separated properly and they're moving to the poles or the opposite ends of the cell properly, so that they'll be genetically identical daughter cells, and that there won't be extra copies of chromosomes or one cell missing copies of the chromosome. So this helps to ensure the chromosomes are attached to the spindles properly so that they can separate properly. So we've looked at both cyclins and as well as checkpoints. So both of those would be internal mechanisms that can help control the cell division. But you also have external influences that can determine whether a cell is going to go ahead and divide or not. Okay? And those may be things that are out of your control, some kind of stimulus, though, that comes in externally that says, hey, the cell needs to go ahead and divide. And a common example here would be wound healing. Right? If I've got a wound, so that means I've got some kind of skin damage happening. So I need to replace these cells. Okay? I need to replace those cells and I need to repair the damage. And so the damage to the skin results in the platelets, okay, those little those fragments of cells that you'll find in the blood that deal with blood clotting. So the skin damage results in the platelets releasing a growth factor. So the platelets release platelet-derived growth factor, which then stimulates other cells, those cells, to start reproducing. Okay? And so that would be an example of an external influence. There was no checkpoint here that said the cell was ready to reproduce. It did not go through any kind of screening process. There's just been some damage, and so it needs to be repaired. Okay, so now that we've overviewed kind of the whole cycle and how it's regulated, let's focus more now on the actual mitosis. And we should be able to hopefully go through this relatively quickly. This should be mostly review. So if you feel that I'm taking for granted things that you should know, or I'm making assumptions, you need to be sure that when I see you in class, you say, you know what, you went over that too fast. We did not know that from our freshman year. We didn't remember it. We need a little bit more help with that. Okay, so when we're talking about mitosis here, remember, like I said, that's going to be the division of the nucleus. And so the goal of mitosis, right, is that we want two genetically identical daughter cells. Okay, so they are genetically identical daughter cells. So they are identical to the parent. They are identical to one another. So for example, if we took humans, 46 chromosomes, that's my parent cell. My two daughter cells also have 46 chromosomes. They're identical to each other as well as the parent. Let's say I had some cell that had 10 chromosomes in it. If it goes through mitosis, still the new ones have 10 cells. They're identical to each other, identical to the parent cell. So there are a multitude of reasons as why cells may go through mitosis in the first place. So unicellular organisms, it may just strictly be for reproduction. Okay, they're a unicellular organism. They need to continue on the species. They need to have babies, so they're going to have babies by mitosis. Okay, multicellular organisms, they have a little bit more, they have a few more reasons why they may do mitosis. And if they're multicellular organisms and they reproduce sexually like most do, then they're not doing mitosis for reproduction at all. They're going to be doing meiosis. But you may have multicellular organisms will need to do mitosis for growth. That's how we think about you as a human. We go from one fertilized egg cell to an organism with trillions of cells. Okay, so that's a significant amount of growth. Okay, they may also need to do some kind of repair or tissue renewal, like we just talked about with the wound. You have to repair and replace those tissues, and that's going to be done by mitosis. Okay, so now we're going to start our list of C words. Okay, remember this is the unit that has 1,000,000 C words in it. Chromatin, chromatid, centromere, centriole, centrosome, chromosome, what a chromosome is versus chromatin or versus a chromatid. Okay, so let's start sorting some of that out right now. Okay, so in interphase, the cell, the DNA in the cell is going to be mostly in the form of chromatin. Some of you guys may remember if you were in my class where you get the pipe cleaner out and we show the relaxed chromatin and then wind it up tight and coil it up like a chromosome. Okay, it's still DNA, it's just essentially how tight it's wound. So the chromatin is going to be the looser form of the DNA. You see this a lot during interphase because in interphase is when transcription is really going to be happening. Remember, transcription is the copying of the DNA to be able to make the proteins. And if it's coiled up super, super tight, then you can't get in there to make a copy of it. So chromatin is more prominent during interphase. Chromosomes will show up during mitosis. And so chromosomes are formed by the DNA just coiling and coiling and coiling and wrapping up on itself. So up here, we're forming chromosomes. And so chromosomes are going to be formed by, first, the DNA is going to wrap around a protein called a histone. So I've got these, what ends up almost looking like a string of beads or a string of pearls. So the DNA has wrapped around these histone proteins. And when it wraps around those histone proteins, okay, it forms what are called nucleosomes. Okay, so I've got this whole string here of nucleosomes that are formed, these beads on a string. Okay, you can see here there's multiple histones there that they have wrapped around and it's coiled on itself. Okay, and then these nucleosomes will then coil even further. Okay, you can see them wrapping around the scaffold, okay, and coiling even further to make the actual chromosome. Okay, and so the importance of the chromosome is it's going to help prevent breakage. So as these pieces of DNA are getting moved around, because you remember the DNA has already been copied, so we don't need access to it really right now as we're headed into mitosis. It was copied way back in the S phase of interphase when it was in the form of chromatin. And so this way it helps prevent breakage as the DNA is getting moved all around the cell, and it also helps ensure that each cell is going to get a copy of each chromosome by keeping them held tightly together like that. Okay, so we're going to go ahead and generate our list of C words so that you can see them and start getting a handle on them. We've done 2 of them already. Chromatin, that loose form of DNA that we mostly see in interphase, okay, versus a chromosome, which is going to be the tightly wound DNA that we see as we go into mitosis. So now let's add a chromatid on here. Okay, let's add what a chromatid is. So, if you were in my class, you may have done the Chromosome Dance, where you stand up and you put your arms and legs over your head and you kind of shake around. If you weren't, we'll do the Chromosome Dance because I think it helps you guys remember it. But we're going to kind of draw it out a little bit here because the confusion is that whether the DNA has been replicated or not, it is still a chromosome. Replicated, it's a chromosome. Unreplicated, it's a chromosome. So let's see what we're talking about here. Okay, right? So if I have one strand of DNA here, this is my unreplicated DNA. Okay, so it is unreplicated So I'd have 46 of these, right? I'd have 23 pairs of unreplicated